Hi, I'm Bill Rapisi, Executive Director of Lymphatic Education and Research Network, and I just want to take this opportunity to thank all of you for joining us today for our Symposium series. I also want to take this opportunity to thank all of you who have become members of LE&RN and help support this series so that we can continue bringing this kind of groundbreaking information to you in your homes and offices. I would also suggest for all of you who have not become members of LE&RN, $5 a month, you can become a member of LE&RN and help support this programming and the research that we do. Thanks very much for joining us, and I look forward to having you become a member.
Okay, hi everyone. My name is Kathleen Caron, and I'm professor and chair in the Cell Biology and Physiology Department at the University of North Carolina in Chapel Hill, North Carolina. And I'm delighted to be here today as part of the LE&RN webinar series. I'd like to thank LE&RN for providing me with the opportunity to speak to you all today a little bit about our research, but also about the basic science research that's going on in the field of lymphatic biology in a broader sense. I'd also like to shout out a thank you to my graduate student, Brooke Matson, who's sitting right over here and watching everything and making sure that there are no technical glitches as we go through the presentation. So what I wanted to do today, considering that there's probably a mixture of audience of basic scientists, clinician scientists, and also the lay public and maybe even patients who suffer from lymphedema or problems with lymphatic dysfunction, is provide a very broad overview, first of the lymphatic vascular system. I think if you're tuning into this broadcast, you're probably familiar with the lymphatic vascular system, but it's always good to have a bit of a refresher. And then to really sort of dive into basic science approaches that really make use of genetic approaches that we can use now in mouse models. And I hope that by the end of this presentation, I will have educated you on some of the tools and tricks that we can use and how these approaches using genetic engineering can be really very informative. Drained out of the capillaries in your capillary beds gets re-uptaken. So that you don't end up just losing fluid into your skin and into your interstitial space. So that's one of the main functions, and the lymphatic system is constantly working to drain that fluid. But there's a lot of other very important functions of the lymphatic vascular system, and that's best shown by how it's connected with the lymph nodes, or these tertiary lymph node organs. And it's a very important conduit and trafficking route for the delivery and maturation of immune cells. So all of our immune cells in our body use the lymphatic system as a highway to get where they need to be, and also to mature and develop the appropriate properties that they do to help fight off infections. And then finally, another important role of the lymphatics, and I'll show you actually some really interesting data from some of our mouse models, is that the lymphatics are critical for absorbing fats and lipids within our gastrointestinal tract. And so as you think about the McDonald's meal that you're eating for lunch, a lot of the fat, actually most of the fat that you're consuming in that diet is being absorbed through the lymphatics that are within your gastrointestinal tract. So really critical roles throughout the body, and very important roles in adult physiology. Now, I mentioned a little bit about how these lymphatics work, and this little movie here shows you that the fluid, which is represented by these little green arrows, is in this space that's in between the blood vessels and the lymphatic vessels. It's sort of the interstitial space, the acellular space between the vascular system. And because of these contractions and these pressures within the lymphatic capillaries, this fluid enters the capillaries, goes through the one-way valves, and eventually, like this cartoon showed, all of that fluid gets returned into your blood circulation and therefore keeps your blood volume consistent throughout the day. Now, in an adult, there's about 120 milliliters per hour of fluid that goes through the lymphatic system. And if you translate that to how much that is in a day, it's about 2 to 3 liters per day. So compare that and think of a gallon of milk, and think of all the fluid that's there, and imagine what would happen then if you have deficits or dysfunction in your lymphatic vascular system. So when it all goes bad, it's the problem of lymphedema, which many of you may be aware of. And there are many different causes of lymphedema, and I've shown a few here on these slides. Primary lymphedema, which is genetic inherited diseases of lymphedema, still considered sort of a rare type of lymphedema, but affects about 100,000 people in the United States. Secondary lymphedema is much more common, and it's an interesting and an unfortunate consequence oftentimes of surgery or radiation therapy, where the lymphatic vascular system has been injured or disrupted. Lymphatic vessels are very difficult to, they're not like blood vessels where you can just see them under your skin, because they're very translucent and they're very frail, and they don't have a lot of structure to them. So surgeons oftentimes will, inadvertently break the lymphatics, or if you're in a motorcycle accident and have a lot of trauma, your lymphatic system may be damaged in your leg, and the repair of that system and the drainage can be very challenging to do. And we see this in this image here of a woman who's undergone removal of a sentinel lymph node, which is a life-saving therapy for her in the treatment of her breast cancer. But as a result of having removed that lymph node, it severs the lymphatic return from her arm, and you can see the lymphedema that develops in her arm. Now, by far the most common cause of lymphedema worldwide is filarial parasitic lymphedema, and this is due to a parasitic infection of a parasite within the lymphatic capillary beds. And in that method, these parasites block the lymphatic drainage and sort of form a functional block to the drainage. And 120 million people worldwide are affected by this type of lymphedema. Now, those are causes that we associate with primary defects with the lymphatic vascular system. But I do want to impress upon you, and I believe Dr. Roxon has spoken on this webinars in the past, that there are many other human syndromes and disorders that are associated with lymphatic dysfunction, and I've listed a few here. And so while there may be other genetic causes for these syndromes, the lymphatic system and its perturbations are prominent clinical features in these types of disorders and many others. And I encourage you to read Dr. Roxon's very seminal work on this and characterizing all of these different lymphatic disorders. Now, lymphatic vessels also play an important role in tumor metastasis. So I did just mention that women in the past who have sentinel lymph nodes removed, and the purpose of that was to try and stop the breast cancer cells from metastasizing through the lymph organs. And so it is well recognized that while tumor cells can metastasize through blood vessels, as you can see in this red vessel here where the tumor cells are metastasizing to a site, the tumor cells can also enter the lymphatic vessels. And in fact, because lymphatic vessels are so permeable and so leaky and so good at trafficking immune cells, it's kind of, they're professionals at doing that, actually metastasis through lymphatics is a really big problem, and is understudied in comparison to the metastasis through the blood vascular system. Now, unfortunately, there are not many treatments or therapies for lymphedema or for inhibiting tumor metastasis. There is a lot of promise with inhibitors to VEGF-C and VEGF-D, and work by Carielli-Tallo's group has very promising results with inhibition of VEGF-C and VEGF-R3. But then in clinical treatment and management of lymphedema, many of you may be familiar that compression garments and massage therapy still remain the most highly recommended treatment to help ward off this lymphedema. And it works, but of course it's a daily regimen, and you really have to keep up with your compression garments and your massage therapy to prevent the formation of that lymphedema. So clearly there needs to be a lot of additional basic science research so that we can identify new pharmacological targets, new genes, and new pathways that might preferentially be able to target and affect the function of the lymphatic vascular system for these very important clinical problems. Now, historically, the study of the lymphatic vascular system about 30 to 50 years ago was largely done in large animals. And I'm showing you some pictures here of some of our favorite large animals in research: dogs, rabbits, sheep, guinea pigs. And Some of the advantages of studying the lymphatic system within these large animals is, in fact, because they're large. So surgical approaches where you can go in and tie off a lymphatic vessel and block its drainage, and then you can study the effects of the lymphatic drainage or the lack of lymphatic drainage. These types of surgical approaches are much easier to do in a large animal because they're larger, and so it's just sort of the benefit of working with a large animal. So there have been lots of wonderful studies, anatomical studies that describe the lymphatic structure, lymphatic flow, modes of lymphatic drainage in these large animals. But unfortunately, a lot of those studies are descriptive in nature, and they don't enable scientists to ask mechanistic and genetic-based questions. And really, we can now do that using mouse models. And so the point, which I hope I will convince you by the end of the talk, is that the mouse model, because of the genetic tractability of this model, does provide us with a lot of benefits to reveal new discoveries in the field of lymphatic research and in general biomedical research as a whole. So some of the advantages of the mouse are that there are a lot of genetic tools, and I'll show you some of those tricks of the trade that we use to tease apart genetic pathways in the mouse. Also, because the mouse is a preferred animal in basic biomedical research, we now have a lot of molecular markers and a lot of reagents that we can use in these tissues. And later on in the talk, I'll show you some images where we can decorate and light up tissues of the mouse with antibodies that fluoresce. And this allows us to be able to better visualize and see lymphatic vessels and study their changes either in normal physiology or in pathophysiology. Likewise, these fluorescent markers can provide us with ways of tracing the cells as they're coursing through a developing embryo. We can also do, because of the complex genetics and the different genetic backgrounds of animals, we can do organ transplantations in the mouse. Of course, the mouse, because it's a small animal, we can house a lot of them, and it doesn't cost too much money compared to sheep that you need a big field to house them in. And they're less expensive to house than these larger animals. And they breed really well. So there's a lot of advantages to using the mouse. But as you can imagine, some of the challenges remain, and that is that they're very, very small, so they fit in the palm of your hand. And so if you want to do a surgery, a lymphatic obstruction surgery on a mouse, that's much more challenging than, say, in a dog or a sheep or a rabbit. So how is it and why is it that the mouse has become such the preferred model? I've touched on a few things, and those of us who do study mice and use mice as model tools, we do like to sort of think of them as little people, because the mouse, although it is a rodent and has a lot of different physiology than humans, we like to think of Stuart Little and Mickey Mouse as sort of being little miniaturized humans, and in many ways they are. Of course, we recognize that they're very different. But across all of biomedical research, the mouse is evolving as a very powerful tool where we can model a whole type of different human diseases. Now, of course, we have to recognize that there are differences, that a mouse is not a human, and you can see this here, and there's a lot of text here, but I'll just walk you through it. So, of course, a mouse is different. It's a rodent, and some of the benefits to using a mouse is that it breeds a lot. So the mouse gestation compared to 40 weeks of a human pregnancy is only 19 to 21 days. They're a multiparous creature, so they have lots of pups, which is good for increasing the productivity of the genetic animals that you're trying to study. They sexually mature very quickly, and as a result of that, you can have 5 generations per year in a laboratory setting. And that's a huge boon to a geneticist who might be studying or trying to expand a colony of a particular gene of interest. Now, the mouse genome is a little bit more compacted than the human genome. They only have 19 autosomes plus an X and a Y chromosome. But interestingly, nearly every human gene, about 80% of human genes, have a corresponding gene in the mouse. So the genes that orchestrate heart development in the mouse are very similar to the genes that orchestrate human heart development, and so on and so forth. And moreover, the genes in the mouse, because we're actually evolutionarily pretty close neighbors, are organized within the genome in the same order that they are in humans. And the reason that this is important is that over the past decade or so, we've appreciated that genes that live in large segments on chromosomes tend to co-function together. And so it's very helpful that the mouse genome has these large areas that are precisely sort of copied from the human genome, and that they're evolutionary conserved genes, and they're in the same order and probably doing many of the same things in the mouse as they are in the human. And this is called synteny, or the comparison or the likeliness between two different genomes. Now, I mentioned that we're evolutionarily pretty close together, so if you think of other types of model organisms used in basic research, let's say Drosophila, like the fruit fly, or C. elegans, the worm, or other models like zebrafish, which is a fish model that's used, those are very evolutionarily far away from humans, about 600 million years. And compare that to the mouse, which is only about 75 to 125 million years. And this image here, which I've taken from Nature magazine and a nice review that they had about the history of mouse models in biomedical research, shows a picture of the common ancestor between mouse and humans. So while this seems like it's a long distance, genetically speaking, the mouse is actually a real treasure trove to study the genetics of human biology. And because of this, when the human genome was first sequenced in February of 2001, the mouse model was the first model organism to be fully sequenced following the human genome, only a short year later. So I think that helps to illustrate that with the power of high throughput sequencing and the genomics evolution, the mouse is really the primary genetic tool that biomedical researchers are using today. And a lot of that is because of pioneering and cutting-edge approaches using genetic tools that we can use in the mouse. And a few examples of that are transgenic mice. So Richard Palmiter's group and Ralph Brinster's group were able to develop a small piece of DNA that would drive the expression of a growth hormone gene in a mouse. And because of that overexpression of this protein product, the mouse grew larger than it would. Now that was in 1982, and it was exciting and extraordinary because of the technology of being able to introduce a new piece of DNA that then would get expressed, made into a protein, and change the physiology of the animal. And those are called transgenic overexpression. And this method is commonly used today, and I'll show you some examples of how we've used that in our lab to study the role of a protein that we're interested in. But the opposite of that is to take genes and delete them. And here, these are called knockout mice. And this is where we can go in and use some genetic tools to remove a gene within a mouse. And the individuals who are credited for developing this technology are Martin Evans, Mario Capecchi, and our very own Oliver Smithies here at UNC. And these three gentlemen were awarded the 2007 Nobel Prize in Physiology and Medicine for this remarkable achievement. And it really is because of the ability to go in gene by gene and knock out a gene and then ask, well, what happened? What happened when we deleted that gene? What went wrong? That helps inform us on how those genes can be functional and important in adult physiology or in development. And this is one of my favorite pictures of Oliver. I actually trained with Oliver as a postdoctoral fellow. He's still at the bench today and a very active scientist, and really an inspiration to many young scientists around the world. So this revolution of these gene targeting approaches and making gene knockout mice has really transformed all of biomedical research. And the lymphatic vascular system is not alone or unique in that example. And what I'd like to just show you now is a couple of images where we've knocked out a gene in a particular gene within an animal, and then you breed that animal and you ask, well, what happens? And in many times, the phenotypes that you see here are shown in the pictures on the bottom. These are now mouse embryos that unfortunately didn't survive to birth. They were either embryonic lethal or died shortly after birth. And you can notice that they look swollen and they have a lot of edema, or there's abnormal blood within their lymphatic vessels, which is something that shouldn't happen. Blood normally isn't in lymphatic vessels. And it's because of this systematic approach where investigators around the world are knocking out genes and observing and discovering the effects that the knockout of these genes have on the lymphatic vascular system, that we've now been able to piece together a very elegant and very detailed map of how lymphatic vessels develop during embryogenesis. And I won't belabor too much of the details of this process, but it is a very interesting process, which shown here on the left, very early in development, a series of genes turn on a group of cells within the cardinal vein and fate them to become lymphatic endothelial cells. So actually the progenitors of lymphatics are venous endothelial cells, the blood vascular system. But these cells quickly change the proteins and genes that they're expressing and become lymphatic vascular cells, which then under a series of different growth factors will proliferate and grow and develop into the mature and adult functioning lymphatic vascular system. And I've listed some of these critical genes here, and what's most interesting is that really for all of these genes, they are now guideposts that we can use to ask whether or not these genes or genes within their pathways are mutated in humans who have primary lymphedema. So many patients with primary lymphedema, the genetic cause of it is not known. And so now at least we have a list of genes that we can begin to sequence and query whether they might mirror human lymphedema or be a primary cause of human lymphedema. And in fact, that has been true for many of the genes that are on this list, that they have been associated with genetic mutations in humans that are causative of lymphedema and other lymphatic disorders. So again, the ability to go in and knock out individual genes has really revolutionized the field and increased our toolkit and the list of genes that are involved with lymphatic vascular function. What I'd like to do in the next series of slides is just share with you our own experience in using this approach and how we have helped to contribute to this effort by identifying 3 new genes that are really critical in the development of the lymphatics as well as the function of the lymphatics in adulthood. And these genes belong to the peptide family called adrenomedullin. So adrenomedullin, it's a small protein, a small peptide that circulates in the blood. It's typically a vasodilator, so it does have profound effects on the blood vasculature. But the gene itself that encodes this small peptide is highly conserved throughout evolution. So starfish have adrenomedullin, and pufferfish, and mice, and primates, and humans have adrenomedullin, and the gene is highly conserved. And usually, when you find a gene that is so highly conserved throughout evolution, you sort of, a little light bulb goes off and says, you know, this gene must be really important if it's so highly conserved. And I'll show you, in fact, that's true, because when we knocked out this gene in the mice didn't survive, and they had some of those phenotypes that I just showed you with that lymphedema. And I'll show you some pictures of what that looks like in the next couple of slides. So adrenomedullin, the peptide, and I show it here in this little red box, has its effects on the body because it signals or binds to a protein called CLR. And CLR is what's known as a G protein coupled receptor. So these types of proteins sit on the surface of cells, and they're ready and poised to respond to growth factors and ligands that get secreted, and in that way they activate the function of the cell to have the desired effect. So adrenomedullin peptide binds to this G protein coupled receptor, which is associated with this other protein called RAMP2. Now this peptide is very important clinically, and if you want to do a little bit more reading about it, you'll probably find a lot of literature showing that adrenomedullin is a really important biomarker in pregnancy and in preeclampsia, as well as congestive heart failure and heart disease and sepsis. And so clinicians are really starting to turn to adrenomedullin as an important biomarker of cardiovascular disease. So on this slide, I'm showing you some of our data where we've now gone in and knocked out each gene of this signaling pathway. So here we have a knockout for the receptor, CLR, a knockout for adrenomedullin, which is the peptide or the ligand, and a knockout for RAMP2, the knockout mouse. And I hope you can observe how they look swollen and full of fluid, and in fact that's what happens. And that's because these genes, which are highly enriched in the lymphatic vascular system, are critical for the growth of these vessels during development. We talked about how these lymphatic progenitors will bud off from the veins and develop lymph sacs, and here's a real picture of what that looks like. So here's the carotid artery through the neck of a mouse, and here's the jugular vein, and here's the lymph sac that has budded off from that. Now a normal mouse has a nice big fat functioning lymph sac, but in these animals where they're missing these important genes, these lymph sacs don't grow. And as a result, you can see that there's a lot of edema. So all of this white space represents fluid that's just ripping apart the tissue. And you can even see this fluid as you look at these embryos here that are full of this edema and fluid. And so now, unfortunately, those embryos do die during development, and so this signaling pathway, as evolution would have suggested, is critical for survival. But using some additional genetic tricks, and I won't go through the details of it, we're able to knock out the gene in an adult animal. So we can overcome that embryonic lethality and then ask what the effects are of deleting these genes in an adult animal. And there's a couple of approaches that we've used to do that, and the end phenotypes are exactly the same, and I just want to walk you through them because they're quite exciting. So the first, of course, is in the eye, and this was a big surprise to us and to many people in the field of lymphatic biology, because a role for lymphatic vessels in the eye had not been well appreciated until just recently. But when we knock out the adrenomedullin receptor in these mice, the first phenotype that shows up is a glossing over of the cornea and this corneal edema that you can see in this bottom panel. And these lymphatic vessels, which are now highlighted or decorated with this fluorescent green marker called Live1, are dilated and very large, which suggests that they might not be draining very well. So there's clearly a predominant phenotype in the eye of these animals. Also, they have lymphedema in their limbs, just like people develop lymphedema in legs and arms. And you can look at these lymphatic capillaries in the tail. They're dilated and enlarged. And these animals develop edema, and they're not able to resolve the edema. If we look at the lymphatic capillaries within their ears, the ears of mice are very thin, and so you can inject a blue dye and immediately see this beautiful spider-like web structure of those dermal lymphatics that I had talked about in the very first slide. And I hope you can see them here, this network of vessels. In the mice that are lacking the receptor, that network of vessels is not well formed, and it's not well functioning, and it doesn't take up the dye very well. And that's because these cells that make up these vessels are disorganized and are hyperpermeable. And so these proteins that are now shown in red typically mark the linings in between the cells and ensure that the cells are tightly packed together. But in this case, on the right-hand side, you'll see that this red protein is all bunched up and inappropriately localized. And so instead of being nice and linear and keeping those cells healthy and functioning, when you lose the adrenomedullin signaling, you get disruptions in this. And ultimately, that also leads to major problems within the intestines. And these images here are showing you a small piece of the mouse intestine that is stained for these lymphatic vessels. And I hope you can appreciate how much of our intestine is actually made of lymphatic vessels. And these little finger-like projections that you see are the little fingers of lymphatic vessels that enter into each of your lymphatic lacteals. And those are the vessels that are absorbing the fat after you eat that meal at McDonald's. And when we look in our animals and we feed them a diet, a high-fat diet, we notice that there's a lot of white, milky fluid within these lymphatic vessels. And the lymphatic vessels in each of these little fingers is large and dilated, again, indicative that there's poor drainage and poor function. And here's the effect of what happens. A normal mouse, if you feed it a McDonald's-type diet, which we call a Western diet, or WD in these graphs, it becomes obese in a very short amount of time. But these animals that are lacking the adrenomedullin receptor fail to gain weight, and that's simply because their lymphatics are not absorbing the fat, because the lymphatics are not working well. In fact, they're losing a lot of protein in their feces, and this is a very unhealthy state of metabolism with these high levels of alpha-1 antitrypsin. Now, so that's a summary of what happens if you lose adrenomedullin in an adult. And you get this limb edema, these big problems with the eye, and of course these intestinal problems and a lack of lipid absorption. And my point that I'd like to emphasize is that these are the main clinical features that are seen in humans who suffer from human lymphangiogenesis. So there are primary causes of human lymphangiogenesis, but remember all those syndromes we talked about. Most of the prominent problems that these patients experience are fully recapitulated in this mouse model. And to our knowledge, this is one of the first mouse models that so nicely mimics and mirrors what we see in human patients. So it provides us with an excellent opportunity to study more about the physiology and potential treatments to improve lymphatic function in these animals. Now, just one slide very quickly, going back to that transgenic approach where you can overexpress a gene. Natalie Karpinich, a postdoctoral fellow in the lab, spent a lot of time developing tumor models where she could make the tumor cells secrete more adrenomedullin. And when she did that, she found that this protein adrenomedullin can drive lymphangiogenesis and make the lymph vessels bigger. Again, they're highlighted here in this fluorescent green. And as a result of that, these tumors within these animals can metastasize to the lung much more readily than a tumor cell that doesn't have adrenomedullin. So if we take all of this together, we know that this new peptide, as well as its receptors, are 3 new genes that we've identified due to advances in the genetic capabilities that we have in mouse models. And we've learned that the expression and function of this pathway is absolutely required and essential for embryonic development, but it's also critical to maintain normal lymphatic function in adults. And of course, in tumor biology, it will be very interesting to start to study the expression levels of this peptide and these signaling pathways in different types of tumors as we start to segregate which tumors might be more prone to lymphatic metastasis versus blood metastasis. And again, all of this has stemmed from capabilities that we have using powerful genetic tools within the mouse. So the lessons that we've learned are the genetics of the mouse has informed us and provided novel new information on new genes and new pathways in lymphatic vascular development. As we identify those new genes, geneticists and genetic engineers can use those very same genes as genetic tools. And so not only are we discovering new genes, but we then can use those genes to do more sophisticated and elegant approaches to target the lymphatic vascular system, either by making new antibodies that fluoresce in different colors or using the genes to mark or lineage trace the lymphatic endothelial cells. And all of that together comes into really addressing the form and the function of the lymphatic vasculature. And some of the key questions I think that remain for the field are now to go in and start to explore the functions of the lymphatic vessels in the different organs. And I gave you some examples of the eye. Again, something that's very new to the field. We never really appreciated the role of lymphatics in the eye until a few years ago. In the intestine, such a critical role that it has in nutrition and overall well-being, and in other peripheral organs and major organs like the lung and the heart. So really starting to explore the role and function of lymphatics within the different organ systems is going to be a critically important area of research for the next 5 to 10 years. And another area that I think really deserves quite a bit of attention is to look at sex differences within the lymphatic vascular system. A lot of basic research will focus on male animals or just sort of the stereotypical animal, but women have, of course, different anatomy. So women have tissues that are very reliant on the lymphatic vascular system, such as the breast, the uterus, and perhaps the placenta. And these are unique organs to women, and we need to be looking at the role of the lymphatic vessels within these organs. Also, it's very clear that the blood endothelium will respond to sex steroids like estrogen, progesterone, and testosterone. And we need to take a closer look at what the response is of the lymphatic endothelium to these sex hormones, because it is reported by patients who suffer from lymphedema that they will have worsening symptoms during their menstrual cycle. So clearly there's clinical evidence that says that during the menstrual cycle, the lymphatic system is likely responding to these hormones. And so what are those pathways, and can those pathways be used or modulated to improve lymphatic function? And then finally, some epidemiological evidence clearly suggests that congenital lymphedema is much more common in women than in men, but the genetic basis for that remains largely unknown. So this is a huge opportunity to ensure that we are diversifying the research in basic sciences to include both sexes as we study the lymphatic vascular system. And some of the gaps of knowledge that my lab and other labs will be working on over the years are to study the different lymphatics in these different vascular beds. Ask what the effects of edema are. Let's say that the lymphatics are injured or not working. Is that really such a bad thing? And what does that edema do to the tissues? Does it cause inflammation? Does it help to resolve inflammation? Does it cause adiposity or fat deposits? And so we really need to learn more about what that fluid, the edematous fluid, is doing to the tissues that are bathing in it. Of course, we need to continue to develop new surgical models and genetic tools, and the mouse is the most powerful one, in my opinion, to do that. But the surgeries are complex, and so just learning new surgical tools is critically important. And I touched a little bit on the role of estrogen and women with the female lymphatic vascular system that needs to be studied. So with that, I'll end the presentation and thank the many members of the Caron lab through the years. That listed here are the current people in the lab, but many people have contributed to a lot of the data that I showed you today. It really is our passion. We're having a wonderful time working on this, and we really appreciate the opportunity to share some of our results with you and hopefully get you thinking about some interesting ideas that you might like to explore in your own research or in your advocacy groups. Now, our research program has been very generously funded by the NIH, the University of North Carolina at Chapel Hill, and recently through the LE&RN Foundation. And we are extremely appreciative to those who have funded us and who help maintain our research activities. And of course, we couldn't do this alone. Basic science research is always a collaboration between many labs at many institutions all over the world. And we're very fortunate to have wonderful collaborators who've helped us with our research. So with that, I'll end, and I believe there may be some questions that are coming in. I'll sit here for a few moments and look forward to hearing some questions that you might post online, and happy to help answer anything that I can. Thanks a lot for your attention, and I hope that this has been a good learning experience for you. Thanks. Okay, so Brooke is telling me here, one of the questions are there any large-scale efforts underway to identify new lymphatic-associated genes? That's a really great question. There are a lot of efforts. The number of genes that we currently know to be associated with lymphatics is not that huge. There's only about a handful, and so clearly we need to learn more. And so what are some approaches that are ongoing? Well, there are lots of clinical repositories and clinical genetic data from human families who suffer from lymphedema, inherited forms of lymphedema. And so clearly they're sequencing the human genome to search for associations and genetic linkages with lymphatic dysfunction and any human genetic causes. But going back to sort of the basis of the presentation and thinking about mouse models, the mouse is a very genetically robust model organism because we do know its entire genome, and in fact we know the genome of many different strains of mice. And so in this way we can actually do some very elegant high throughput genetics to start to screen for genes that might be involved in lymphatic development or lymphatic function. So, for example, instead of trying to look into a crystal ball and guess which gene might lead to a lymphatic defect, we can go in and randomly mutate the genome and then look and see whether or not a mouse has lymphedema, and then work our way backwards to identify what that random gene was that got mutated. Some other approaches are to knock out every gene in the mouse genome. And this huge project, which is an international consortium between the United States, Europe, and Japan, is to, once again, knock out every gene in the mouse genome and analyze its phenotypes. And that project is called COMP, K-O-M-P, the Knockout Mouse Phenome Project. And so they are systematically going through and reporting and making freely available to the community, and this is all freely available information, the phenotypes of animals as they knock out gene by gene. And so as an interested investigator, I can go into these databases and look to see if there are mice that may have phenotypes like I showed you with that edema. And maybe those mice would be interesting for me to go and study. And so again, these are freely available resources to the biomedical community to try and identify new genes and pathways. So there are a lot of efforts to identifying new genes in a high throughput way. Okay. Brooke is reading me another question. Just a moment. Let's see. Do you think adrenomegaline could ever? Oh, okay. Okay, I see it. So another question is, do you think that adrenomegaline could ever become a therapeutic treatment for lymphedema? So this is a great question, and I actually get asked this question a lot, and I'd like to thank all of you who reach out to me and the members of our lab to ask this question. I encourage you to please continue that dialogue with us. So there are efforts to use adrenomegaline in the clinic, and there are some studies in Japan, some clinical trials in Japan, not for lymphedema, but for heart failure, and they've had a lot of promising success. So I think the important take-home message is that this peptide can be a therapeutic, and it's been shown to be a therapeutic in other settings. Now, how or what it might be doing to the lymphatic vascular system is still a question that we need to resolve and figure out. And a lot of our basic science efforts are geared towards answering those types of questions. Now, the other neat thing about adrenomegaline compared to a lot of the other genes that I showed you today on those lists is that it binds to this special type of receptor on the cell surface called a G protein coupled receptor. So it turns out that this class of molecules called G protein coupled receptors make up about 40% of all drug targets on the market today. So pharmaceutical companies really love G protein coupled receptors because they can make small molecule inhibitors and activators, and they can very finely tweak the pathways of these receptors using small molecules and pharmacological tools that they have. And so it's a really nice opportunity to have this signaling pathway characterized for the lymphatic vascular system. There are not many currently known G protein coupled receptors that play such important roles in the lymphatics. And so we're hoping that our research reaches out to those in the pharmaceutical industry as a potential candidate for them to target and develop small molecule therapies against. So, okay. So another question, does your lab collaborate with clinical researchers focused on gene therapy for treatment? So again, a wonderful question. So we do collaborate a lot with clinical researchers. We always learn a lot from the MDs, and we really benefit from having that type of integrative research approach. And so it's always important to keep the clinical outcomes at the forefront of everything that we do. And so we do collaborate a lot with clinical researchers. However, right now, we're not focused on gene therapy because, to my knowledge, there isn't yet a human condition that's been identified where with a mutation in either adrenomegaline or CLR or RAMP2. Now, of course, if a family were to ever present with such type mutation, then of course that would be, a prime question to address right away with clinical geneticists and gene therapy folks who are, ready to target those with tools that they have. That's a really great question. Thanks a lot. Okay. Well, I think that's probably all the questions we have for now. And again, I'd like to thank you. I'd like to thank Brooke again for being here and looking out for any technical glitches. I really appreciate the time you've spent with us today. And I hope that you've learned something from this webinar. And thank you again for your support of LE&RN Research Foundation. We're indebted to their enthusiasm, support, and advocacy in the field of lymphatic research biology. Thanks a lot. Take care.